got some really neat slides for you here, and I think it's going to show you at a very fundamental level how phosphorus changes ZSM-5. And of course, it's natural to understand that it's going to lead to new developments and new innovations. The first technique I want to talk about here is something called atom probe tomography. So big name. And let me walk you through the steps of how this works. It basically starts with a ZSM-5 crystal, which is shown in picture A. But in order to analyze it, we have to chop out a tiny portion. And in picture B, what you see there is the crystal after it's been hit with an ion beam that carves out a little lamella or trench. It's right there in the middle. And that, before we carve it out, we put a platinum and carbon cap on it that basically kind of protects it. In picture C, you see the scientist using something called an omniprobe to lift out that lamella of the crystal. And then in picture D, they're inserting it and setting it on top of a silicon micropost. So the set is done by picture E. And then the way this technique works is you really have to have a needle-shaped specimen. That's required. So the next step is to take an ion beam and do annular milling around it and hone it back down into a real nicely shaped needle again. All right? After that, they put it in another special apparatus and you start hitting that needle with a laser. And each time you hit it, you start popping atoms off. One atom or a few atoms. So the atoms will then move and hit an XY detector. Okay, so we get XY position. And if you know the time of flight, you can get the third dimension. And if you have a mass spectrophotometer, you can get atomic identity. The end result is that you can put together a picture like that on the right, where you fully map out the location of the individual atoms. That's pretty neat. So let's see here if this movie will play. Yeah, so we've got a movie here that kind of spins it on the right-hand side of the figure. And we can all look at that and say, oh, that's pretty neat. It looks like the aluminum in blue and the phosphorus in red are relatively homogeneously distributed. And we can actually prove it. There's a mathematical technique called spatial statistics, and we can put together what we would consider a random distribution and plot it. You see those in the middle. And we can compare the experimental actual data to that theoretical random distribution. Plots line up pretty nicely. So we know in this particular case, we phosphated the crystal in such a way that we got a nice even distribution. So that's very insightful. But we want to know more. Okay, the phosphorus is distributed well, but how is it actually bound to the framework? What is it bonding with? Oxygen atoms? Something else? And the team went and did some NMR work to really look at that. And there's quite a bit of information on this slide. I'll try to walk you through it at a high level. The first thing I want to start with is the parent ZSM-5 sample that's in the lower left-hand corner. No phosphation. And here we see expected signatures. We have tetrahedral framework aluminum with that big peak at 53.4 parts per million. And then we also see some octahedrally coordinated aluminum in the extra framework position. Now, even if you don't know anything about NMR, you can look at this next one on the right in red, right here, and you can tell these scans are dramatically different. And that's what phosphation does to ZSM-5. It makes a huge impact. I put the arrows on the upper left-hand corner. That phosphation causes the aluminum to move from a clean tetrahedral arrangement to one that's more distorted. And we also start to get different things like 5-coordinated aluminum. The next one is the blue scan on the right. Very dramatic shift again from that extra phosphorus. It makes you understand that it's really an optimum with that phosphorus loading. Here, that tetrahedral peak is basically gone, and now we have a peak that's that's quite big, and we understand that that's associated with octahedral aluminum, and it's probably got a fairly distorted electronic cloud around it from the phosphorus. On the right-hand side, we've got the phosphorus 31 NMR data. It shows us aluminum-bound orthophosphate, some extra framework, alpo as well. And look at the dramatic shift again when you go to the high phosphorus loadings. You've got this shape, the peak is of course very dramatic from this one, dramatically different. And one thing here is now we've got a whole new set of species showing up. We've got these silicon phosphates. We've got a silicon orthophosphate, a silicon hydrogen phosphate monohydrate, and a silicon hydrogen tripolyphosphate. So very, very different materials showing up with that high phosphorus level. Okay, so proton NMR data will tell us some more about the structure as well. I set the slide up in kind of the same way. We've got the parent ZSM-5 sample on the lower left. It's showing us hydrogen signatures consistent with external framework, aluminum OH, Bronsted acidity in the zeolite, and water on the Lewis acid sites. Then we've got some really interesting things that show up on the next two samples, okay? And this is the most important one, really. There's peaks on both phosphated samples that are consistent with the formation of POH species. That's an entirely new acid site that's being formed in the ZSM-5 when it's phosphated, okay? And we'll talk some more about that and where it's located as I finish the next couple of slides. On the right-hand side, we've got cross-polarized data between the 27 aluminum and 31 phosphorus. And what that basically tells us is that the aluminum and phosphorus are in close proximity. So it's going to be things like external framework, ALPO, short chain middle groups, and free chain groups, okay? All right, so now we have an idea of the actual atomic bonds and species that are in this ZSM-5 that's been phosphated. The next question is, where are they in the crystal, and where are they located? And that's something we can get out of high-tech X-ray diffraction. So this gentleman here in the picture is Dr. Elko Vogt. He's one of the key participants in this. And on the left-hand side, he's standing at the ID22 beam line at the synchrotron facility in France, and he's preparing a sample for very high-resolution XRD analysis. And then on the right-hand side, he's in front of the neutron diffractometer at the University of Delft. Okay, so this is a very complicated XRD plot full of a lot of information, but I'll walk you through it and give you the highlights about it, okay? The first thing I want to mention is that we've got the normal signal of intensity on the y-axis, but instead of 2 theta on the x-axis, we've got something called Q. And the reason we do that is just because it's not dependent on wavelength. So you can take different techniques like neutron and X-ray data and compare them, right? Even because Q is independent of wavelength. Now, standard XRD scan for ZSM-5 is going to be kind of like this orange one, right? XRD scan, copper-based radiation. This is what you're going to see pretty often out of most routine laboratory instruments. And the thing that'll jump out for you right here is that a Q value above 3, there's not a lot of information left, right? What that means is you're missing the short-range interactions that are really relevant to the small atomic configurations. Now, these high-quality X-rays that are available from that French facility already are showing us a lot more information at a Q value above 3. And look at the neutron diffraction data. There's all kinds of information in there. And it might just look like noise, but it's far from it. It's actually telling us key things about the average interatomic distances and the structural disorder and the levels of coordination. And we can actually decipher that out. And the way it's done is by transforming the data and starting to do a little bit of guesswork, okay? This particular plot right here is something called G, or the pair distribution function versus R, which is the interatomic distance. That's basically a Fourier transform of that scattering data. And that tells us all the detailed information about atomic configuration. We can start assembling a crystal and creating a theoretical PDF. If it matches the experimental one, great. We know exactly how the atoms are oriented. If it doesn't match, we change it, and we keep trying again until we get the crystal arrangement set up. And the team did that work. The experimental and calculated PDF are here, and they look pretty close. So let's see how this crystal is actually oriented and set up. This is what it looks like. On the left-hand side, it should look familiar. Standard ZSM-5 with those nice parallel pore channels. On the right-hand side, you've got the phosphate-modified ZSM-5. And I know you guys are kind of far away from it, but the phosphorus are the yellow atoms in there. Hopefully you can see then that you've got those POH groups oriented in the pores substantially. That's very interesting to see where they position themselves. That means that those acidic groups are available to participate in reactions. And you can also see that they're perhaps even blocking the pores. Now, I think all the suppliers have speculated for years that phosphorus blocks pores. Here's some real fundamental evidence that says it happens, and this is how it happens. So that's pretty insightful, and I think it's really interesting, and it's going to lead to a lot of new innovations. The last reality check on this is going to be through something called quantum mechanical modeling. fancy stuff. The real point here is that even if we speculate that all these structures exist and all these bonds form, wouldn't it be nice to prove through some sort of thermodynamic analysis that all this is feasible and it's not some incredibly endothermic reaction that can't happen? And it's nice to look at this. If you see on the right-hand side a number of steps, they're exothermic steps, and we know that this phosphation and integration of phosphorus into the framework is really possible. Okay? So just a little bit more about these pictures. This is a standard cutout of a ZSM5 unit cell here. On the right-hand side, you see the first step in modeling, which is where you have a heat treatment and hydrolysis, and you generate some partially dislodged aluminum. And then on the bottom two figures, you see 1 and 2 phosphorus atoms inserting into the framework. And I was really interested to read the team's work on this because it turns out that there's some things that just really probably don't happen. Give you a great example. A fully dislodged aluminum atom that creates a silanol nest. Turns out for phosphorus to integrate there would be very, very endothermic. It's not going to happen probably. So what's really the case that happens most likely is a partially dislodged aluminum, then phosphorus moving in, joining the framework, and healing it and participating. Okay, quick snapshot then of the model. We have basically a situation then where we have a ZSM5 with a number of silicon atoms. You have aluminum atoms in here as well. It's been heat treated. We've got some dislodging of some aluminum, and we've got some partially dislodged aluminum species. We do the phosphation, and as we said, you can create some polyphosphate species, some external alpo species, and then we've also got phosphorus integrating into the framework. You get a very high phosphorus loadings. You can actually get entirely different species like these silicon phosphates that we discussed earlier. Okay, let's shift gears and talk about a couple of products that are out right now. I want to talk